All right. All right. Well, thanks everyone for joining us today for our Knowledge Portal Network webinar series. I'm going to kick it off as Noelle mentioned, and then I'll hand it off to her. But the first thing that we wanted to touch upon were some events that are going to be going on in November in the world of the Knowledge Portal Networks. And our first one that we'll be attending is the American Society of Human Genetics, or ASHG, in Denver, Colorado in November. So if you will be attending, please come and attend our ancillary event. This event itself will be devoted to new features that are within our resource, readouts from our recent widely attended predicted effector gene workshop, updates from novel approaches for gene prioritization, and more. We have talks that will feature some of our consortium members, including Maria Costanzo, Jason Flanick, Laura Scott, and a few others. If you're going to be there the entire week, we also have our traditional exhibition booth. So our booth is number 571, so keep an eye out for that. We also have a brand new update for the portal that we will be showcasing at the booth this year, so you'll have to come by to see it for our first reveal. So we hope to see you there. Okay. So our second event that we have is for the world of the CFDE, or Common Fund Data Ecosystem. And so this is another award that we are working on for building a knowledge center. And we are also going to be at ASHG this year. We will also be hosting an ancillary event and exhibition booth. So we have double the fun with events that are going on in ASHG. So our ancillary event is going to be a perspective for human genetics, and we are going to have an overview of what the CFDE is if you haven't seen it before. We'll do some other conversations around the Knowledge Center itself that we've built, and also have some scientific stories that are a little bit different, and include some of the other organizations that are involved with the CFDE. And one to note is MotorPack. And so if you haven't heard of them before, you can kind of learn about what they are and what they do and how they all come to play with the Knowledge Center. And if, again, you're going to be around the entire week, we'll also have an exhibition booth for this. This is actually going to be next door to our booth that we have for the Knowledge Portal Network itself. So you can come meet us and other members of the Common Fund Data Ecosystem to talk about it. We'll also have some other attendees from the ecosystem itself, including members from the NIH, our collaborators at the Workbench, and we'll also have GTEx and MotorPack there as well. We'll be doing demos and flash talks and things like that. So if there's anything particular that you want to come see for that event, keep an eye out for our different hour sessions that might be involved. We'll be promoting those as we get closer to, but we're hope to see you there as well. I'm not going to go too far into this because Noelle is going to spend most of her time on this, but we have a production release that's upcoming in October that will coincide with ASHG. We'll be unveiling all of this for the first time there. And like I said, Noelle will touch upon this more, but this is just a little preview. For the update, we also have some new data and UI that will be available for the aging KP and neurodegenerative disease KP. So keep an eye out for that as well. And as I had mentioned, as we are involved in other NIH efforts beyond human genetics and genomics, we're going to be discussing this at the ASHG, as I said, but just wanted to bring this to everyone's attention that we're also doing a October release for the Knowledge Center, as I mentioned a little bit before. So we'll have updates to this UI itself. It's basically a single entry point for all things Common Fund. So with that, this is an early product, and it may be of interest to you if you're studying human disease or specific genes. Essentially, our goal is to elevate awareness of key resources that are within the CFDE. And we're also going to provide workflows to address scientific questions, including those for differential expression and gene set analysis. So if you're interested in either of these, this will be a great time to also coincide with our updates. One other thing that I wanted to highlight was we do have a Common Fund Data Ecosystem public webinar series that's very similar to our Knowledge Portal webinars. And so with that, we have our upcoming one on Friday, October 25th from 1 to 2 p.m. And we're actually going to be featuring MotorPack, which is the Molecular Transducers of Physical Activity Consortium. They're going to go over their data hub, some major release updates, and what their pipelines and database structure will be. So if you're interested in attending, please let me know. I can add you to our mailing list for this, and we hope to see you there. And again, if you can't make this and you're at ASHG, MotorPack will be there too. And then lastly, I just wanted to highlight our upcoming meeting dates. Our next date that we have is November 14th, and we'll be doing a focus group session. And then December 12th is our last KPN webinar for the year. So these are our upcoming 2024 events, and we'll update you with the 2025 as we get closer to the new year. So with that, I'll hand it off to Noelle.
Any questions for Mackenzie too? While I'm getting my slides up and ready. Thanks, Mackenzie. That was excellent. Sometimes I don't appreciate the full breadth of the things that are going on in our universe, and it's really cool. All right. So today I'm going to, I was thinking as Mackenzie was saying that the end of the year is coming, I was thinking this means 2025 is coming. And in 2025, the portal, the type 2 diabetes portal turns 10. So that's wild. So appreciating that makes us feel a little old as a resource, but also really speaks to the theme I want to talk about a little bit today and sort of talk about some new adventures, because I think, in the last 10 years, when we started this endeavor, there weren't things like portals. There are very few, and now portals are everywhere, right? It's this resource and that, and if you're doing research, you probably use many of them in your daily life. And so I think that's awesome for the field, because I think the goal is to narrow the gap between, discovery and actually better therapeutics for our research community. And I think using online resources and the data we've generated in this space for human genetics and genomics is a powerful advance of our field. So it's quite exciting to be part of that. But on that note, I really want to talk about today in the context of our Common Metabolic Disease Knowledge Portal, where we're headed, and some new endeavors for us, and some new offerings coming, and just tease you a little bit with it, but also talk to you about the broader picture about why we expanded beyond just diabetes. And then common metabolic disease for human genetics and genomics into the Knowledge Center for Common Fund. Why we as a group between Jason and me and Kyle Galton, thought about that. Why did that matter to us? Because we didn't want to spread ourselves too thin, but there's like a central theme to all of it. So I'm going to start to bring that in as I talk today a little bit. And the reason I do that is because I think resources need to address, if a successful resource has to have two elements. It either serves a community or group of researchers, or serves a scientific gap. And you get really lucky if your resource over time sometimes does both. That's the goal, right? We've been very lucky, I think, with diabetes, the diabetes portal, to do just that over time. And I think that we try and keep that focus as we grow. And that's hard, I'll be honest, because you feel like you have your hands in too many places. So you try and focus on what you're trying to do for the community, whether it's a community-driven resource or a scientific gap. So the scientific gap, I think that we really enable with our Common Disease Knowledge Portal, specifically for the CMDKP, is to make the job of a human geneticist or someone who's studying a gene or a trait a little bit easier in trying to determine which genes you might want to study, or the role of evidence for a given gene in disease. And we do that by making a lot of the work that all of you who are joining this call are experts in easier. I'm having trouble, actually. There we go. So that's our goal, right? And if we enable this serves a scientific gap. But this is done because the community data behind this is like pivotal to its success, right? And so our core competencies we really rely on are, we rest on the community to provide the right data, the right requirements for what we build. And we build a platform that is flexible to those changing requirements over time. And we deliver knowledge, not all the raw data, not all the underlying things, because I think other resources do that way better, but we deliver summary representations of those. So that's our core competencies. And that's the theme and the spirit we take it on in each endeavor we take. forth. So today, a couple new adventures. This is really exciting because I think it's a broader reach across NIH and industry that I think is going to be very fun, but has a very specific early mandate, which is great. It serves a simple gap. And then we'll talk a little bit about some new offerings. So I don't know if you knew this, the AMP, the Accelerated Medicines Partnership, which is the funding mechanism that started the portal under NIDDK and the FNIH, and also expanded to other disease areas, had its 10-year anniversary. And there are many different AMPs in other disease areas. There's an Alzheimer's one, there's Parkinson's disease, there's a new heart failure one. You can see them all here. And they all have, a commonality, which is they're trying to they're public-private partnerships. They are aiming to generate data and resources to make better therapeutics for these diseases, right? That's really their goal. And what's excellent about them is they're really expansive. They have industry partners, they have nonprofit partners, an incredible investment from both the NIH and industry, and the FNA sort of stewards that. And it's really been, for me, just an exciting component activity to be around, particularly learning about the other AMPs over the coming years. But as a result of that, there's an exciting new initiative that's percolated over the last couple of years, but really in the last couple of months, which is called the AMP Systems Biology of Inflammation, or SBI as they love to call it. And the sort of the themes behind that is that there are many AMP programs that the diseases could be considered characterized by inflammatory processes. Also, their goal is to, identify, the goal of this initiative is to identify shared and distinct mechanisms across multiple diseases to try and understand how inflammation might contribute to these diseases more broadly. And because many of these programs have been around for 10 years and generated a lot of data, they're hoping to integrate the data sets across these to answer scientific questions. So many of us were involved in the design phase. This is the folks in government and private sector, but they also engaged us as scientists, as leaders in the various AMPs and disease areas in the design phase. And this is really a fun process. So I'm representing a lot of people when I'm talking about this who have worked both at industry, pharma, and academia. But it's led to this AMP Systems Biology of Information. You can read about this on the FNH website or NIH, but this is the program at large. And its overview is to really, use the existing data and tools within all the AMP current platforms to enable discovery, generate cross-cutting resources, and identify commonalities and pathways, really thinking about inflammation. So how to do that? Well, there's going to be a big process, be a lot of funding opportunities, but really one of the first thing to do is wow, all these AMPs have platforms to make their data accessible. What's awesome about the AMPs is one of the mandates was, these data have to be accessible to others. So they can be useful, right? And so I'm going to talk a little bit about the different ways AMPs have done that in a second. But the folks in this list, and I might be forgetting some, but we were very lucky. Jason and I at the Broad for AMP CMD, we're lucky to be involved in this really cool group of people who are trying to figure out, what is the platform that will help create interoperability between these AMPs. This was a fun project to be part of. And so some of the things we considered part of this working group, we considered things like governance, data security, infrastructure, and actually the data itself, what people want to see. And we did actually a really kind of comprehensive landscape amongst internally to our AMPs and working together as a group to determine the different types of data across the different AMPs. And just to pull out some themes from this, you'll notice there's some themes across all the AMPs. Raw and processed genomic data, particularly around transcriptomic data for different tissue types, and it's all in human for the most part. And the target tissues are a range of target tissues, as you can see here. We have a lot in common metabolic disease because we cover a lot of disease areas, which is excellent. And so some thematic things. One of the things that differentiates us is we also do, in addition to a lot of the transcriptomics and genomic data, there's a lot of genetics. So we're the most heavy genetics of the AMPs, just for perspective. But each of them have one thing in common, right? They all have a very nice and elegant infrastructure to deliver data. And they have portals and resources. They're all in very different states of development. Heart failure is brand new. I don't think even there's data accessible just yet. They're building it through this HeartShare program. Some of them have been around for 10 years. And each of them have a very different approach and a community and ethos. And that's kind of exciting, but it's also challenging to figure out how to bring all these together, right? So their governance structures vary. We are the most open because we really focused on summary level statistics and that upper distillation of results for the community. But we do, as part of all of these, have individual level data, particularly around the genetics and the genomics data, I mean, the genomics data specifically. We make those accessible to others. So that's good. But each AMP has a different, path by which you access, whether it's internal to the consortia, whether it's a permission-based, whether it's through an external party who grants you access. Those challenges all were mapped out in the past couple months across the AMPs. And the programs themselves have had, because of their communities or because of their specific data types, they have had different ways to, present these data and resources to users. So AMP-AD sort of has two major things. They have the AD Knowledge Portal and what they call Agora. So the AD Knowledge Portal is really about, I want to get access to the raw process data, and that's very tiered based access, and that's through Sage Bionetworks, for example. Then they have Agora, which is a sort of a window into their curated top XY genes, a number of genes that they believe are the candidate genes for Alzheimer's disease, for example. Then you have the CMD portal, which is really focused on all the process data, analytical results, curated results for users, accessible to all the time, right away, sometimes before publication. But we put all of our genetic data in EGA and dbGaP, just because by the nature of our consortium. And that's fine, because we ourselves as a consortium gate the access to that. So that's just give you the compare and contrast to that. As a result of that, you have varied users at the other end of these portals. You have folks who are experimental biologists, and you have computational biologists. And that's sort of one big class. I'm forgetting a lot of other people, right? There are clinicians, there are drug developers, but some people like to think about things as, what do I do with the data? Some people want to use the data, if you know what I mean. So those are the sort of the broad classes of people. And then we think about different stages of the data, right? There's the raw data. Those are the FASTQ files and things like that. Then there's the process, the metadata, the transcript counts, the variant calls. And then there's a presented analysis. Here's an example for Alzheimer's disease that we put forth, just to give you a sense of the span. And truly, these analytical results are super easy to make accessible. They require, though, a major infrastructure to curate, process, and present. These are sort of in the middle ground, and many people want these because they at least can get to this more easily. These are harder because they have protections to them, right? But all of the AMPs have these in common in some way, shape, matter, or form across different tissues. So the NIH started a new program called the Venture Program. You can read about it online. I'm not going to describe it. I'm not from the NIH, nor do I know everything that they're doing. But one of their key programs, I think there were two in this first round, was this CysBio. So this was from the AMP SBI initiative for systems biology of inflammation. They said, well, the first thing we need is a platform to bring all the AMPs together. And a lot of us were on the working group like, oh man, another platform, and how is this all going to work? But we had done the work of the past couple, almost a year now, to do the governance assessment, to look at the landscape, to work together, to get to know each other. It's been a great experience for, I think, Jason and I and our team, because we've learned from the other AMPs over the last year, which has been exciting. So they put out a call for an RFA to build this thing called CysBio, which is the Common Fund Systems Data Platform, specifically for all of the AMP partnerships. And one of the key core components was, the disconnected data to answer new program initiative will create a centralized portal and tools to allow researchers to search data across existing platforms, empower reuse, reanalysis of current scientific questions. So that's really the goal. So as a result of that, we were very lucky. So many of us, there were people that put in applications, and we wanted to, work. We as an AMP CMD Jason and I didn't want to put in a prime proposal because we felt like that would be a lot of work to take away from our AMP CMD. But we were very lucky to partner with Verily, who actually builds the AMP PD resource. And just yesterday, I don't know if you saw this, there was an announcement that a Verily-led consortium was awarded to build SBI. And AMP CMD is part of that, I'm very happy to say. We are funded along with several other groups. We're funded with other people. We're funded with Verily, Data Technica, Technome, Sage Bioworks, and at the Broad, and Vanderbilt and Northwestern to build this CysBio platform. So what's exciting about that is, A, we as AMP CMD have a seat at that table because we are part of it as leaders to drive the requirements. But B, I think it's a testament to the AMP CMD portal because our particular piece, we are funded to build the actual visualizations. So I think one of the things that we play to our strengths, because our strengths are that we build visualizations to summarize, whereas Sage and the other teams on that are really good at the underlying, particularly Verily, in terms of its actual ability to store and process this raw data. So this is kind of an very exciting opportunity. So some of the elements, and this is back to the science, which is actually the fun, and the tidbits from the RFA that I wanted to pull out. This is specifically from the RFA. I thought this was useful because I think it speaks to some of the things we want to do scientifically going forward as a community, is that, the goal of it is to explore mechanistic hypotheses across different tissues and different diseases, right? So all of the different diseases in AMP and the different tissues. Allow comparative analysis of omics data from the different AMPs, develop an infrastructure to present an integrated view of human disease. That's what the initial platform will help initiate. I don't think we'll solve all the problems right away, right? This is the beginning phases. It will allow queries of specific genes, molecules, and cells across these diseases and tissues that you couldn't do in one single AMP right now. And then finally, the initial, the minimum viable product that the program is meant to do is to build basically a portal that allows you to report relative gene expression on a per sample basis for bulk RNA-seq within each cell type, and different tissues across the AMP programs. So this is a pretty audacious goal, and we're just at the beginning. We just award yesterday, but I think the sort of the takeaways for you as a community is that it's a way of not building another brand new resource, but really leveraging the skills and the strengths within all the AMPs, because on the award that Verily is leading, you have all the AMPs represented already. So the scientific sort of brain trust, if you will, from the AMPs brings that community together, which is excellent. And you have the technical muscle of Verily and other technical components to bring that infrastructure. And we have different opinions contributing different parts to deliver this. So what's excellent is you'll have one portal at the end of the day to explore all of the AMP data, but each of the respective portals will still maintain who they are, their own governance for the data they're generating, and also their own portal. So our vision really, and this is something, we have to work out over the next 3 years, is if you are an investigator and you are an AMP CMD investigator, for example, you'll be on our portal doing your thing like you usually do, I hope, but also you will have a window right there in your AMP CMD portal to the AMP SBI. Oh man, gosh, I can look across all of these other tissue types in other disease states, perhaps, right? That may not be as interesting to you if you're studying diabetes, but you would like to know that. But other tissue types that will come together as well as that may be relevant to your disease. So this reciprocal exposure is really an exciting development. And we're going to make it so that it's really easy. If you live solely in AMP PD, you're going to know about SBI because we're going to bring it to that community. But also SBI as a portal will also be a standalone entity. And that's really the vision. And so I just wanted to welcome you all as members of the community. This will be something that we'll be talking about. What are the requirements? What does this need to do? How does this differ from, visualizations we already have? How can we take visualizations we do have or that other researchers have done well and make those integrated in the platform? So that's really exciting. We're very happy to be part of it. And you'll be hearing more about it as we go. So any questions or comments about that? That was just more, teasing for the news. So new offerings. That's sort of my next feature for you all. How are we doing on time? Excellent. Okay. So for this, I wanted to think about back to the Common Metabolic Disease Knowledge Portal. And, again, back to the theme of we need to serve a community and serve a scientific gap. I think for the portal, we've done a pretty good job of serving the community for genetics, right? If you are studying a specific disease and you want to know what human genetics tells you about it, for common metabolic disease, you come here. If you're studying a gene or variant or a region, I think, and you want to understand its role in disease, for human genetics, you come here. And it's pretty easy to navigate, and that's been useful and good. However, I think we've been hearing more and more that we want to see users want to understand, well, if I'm going to study a gene or I need to know what tissue to study it in. I need to know its role. And even beyond the tissue, I care about the cell type, right? And so, we've been hearing this for a while. Our field has done an incredibly good job, not our field, other people's field, in the single cell space and the transcriptome space has done a great job of figuring out how to generate more data in this space. I'll talk about that a little more. But, we thought the first place to start to step out of phenotype-based visualizations and presentation of genetics results and genes and region is to shift towards, well, talk to me a little bit about the tissue. So in our last release, and I think Maria talked about this at a webinar, we talked about presenting results as a tissue. So we have that in a very early version. So that's just the beginning of starting to get down that path of like tissue presentation, the genetics in the context of a tissue, right? So the first thing we started was like, let's just present what we know about a tissue, just to start with that. So for example, right now, if you go in the portal, it's actually here today, you can see for adipose, for example, the top gene expression for adipose tissue. For this gene expression data, we complement, GTEx data is integrated and represented here, along with AMP-CMD specific gene expression data that's been contributed and other stuff that Kyle's team at CMDGA has aggregated. So just, you get the summary of the top genes and expression values for that tissue. So it's just a visualization, right, for now, right? But it's all harmonized and presented to you in one place. And below that, you'll also then see the global enrichment, which is actually a useful analysis based on mixed answer stream for this particular tissue. And you can see the biosamples, the underlying data for that, and the phenotype. And so this is just one beginning of how we're contextualizing human genetics in a tissue. But that's really limiting, right? That's only goes so far. How do you go beyond that? Well, I think we've been hearing for a while that this is where we want to go. Single cell presentation. The tissue doesn't really help. You need to know about cells, right? You need to know which cell is expert. How does it change the abundance? All these questions. And particularly within our consortium, right? We hear this exact thing. We are generating so much of this data. Where is it all going? Like, how do we use it? Like, our consortium itself is generating this. There's a lot of awards from the FNIH, specifically in common metabolic disease tissues, to develop, data sets in disease data, not across these different RNA-seq single cell RNA-seq experiments, and make them accessible. Right now, we don't make them fully accessible. We can store them, but we don't really present them in an elegant fashion. How do we use them? And more importantly, how can they be integrated with genetics? It's simple to visualize them with UMAP plots and dot plots. That's a start. But how do you use them to answer scientific questions around, variants and things like that? So this is a work in progress and touches upon so many other resources that are tackling the same problem, including the CISBio initiative. One of the core goals is gene expression in the context of cell types, in the context of genes. So we're trying to not build a bunch of resources, but trying to systematically address this question across different contexts, depending on the way that you approach your scientific questions, right? So the field is doing a really great job. This is an excellent review that just came out that sort of anchors us to, this single cell genomics is meeting human genetics. There's a lot to do here, a lot in this space, a lot of challenges, but I think we're meeting that space much better. You also have folks in our community, particularly around common metabolic disease, who are building integrated maps, who are taking this data, building a map, defining the cell types for us. We have folks like Evan Rosen's team doing this comprehensively for the adipose tissue. You're seeing this all over the place. But what about that in the context of human genetics, right? So bringing that all together is something we're focusing on. So you have these beautiful presentations of the UMAP plot, the differential, the different abundances of cell types, and the visualizations of this results for a gene and a different condition, right? So this is like sort of the model we want to get to. So to build that, we need to get all the data together into CMDGA, create what they call embeddings, or ways to actually make the data sets processable, create integrated maps across the data sets we already have, community-specific data sets. So gather those community data sets that are generated across pancreas, for example, across liver and other common metabolic disease relevant tissues. Bake in the reference resources like HCA, HubMap, who are doing this at scale and have incredible resources and tools and domain knowledge. Engage them, work with them, not rebuild things that they've already done, but also build tools to dynamically query these by age, sex, state, and allow you to view that with different changes. So you can address scientific questions, right? And these questions are things like changing genes expression, what genes are preferentially expressed in my favorite cell type, and more. So, we have two objectives over the coming years to sort of bring this together, just to simplify things for us, create comprehensive integrated maps of key tissues involved in metabolic disease. So that's one thing that we'll be doing as part of the common metabolic Disease Consortium for AMP in our last 2 years, develop intuitive visualizations. Now, what I mean by intuitive, this is the word. We need your help here. What's intuitive? What's useful? But in a moment, I'm going to show you a very early version of that, just to, see if this gets you thinking. Because, let's talk about that. Actually, I have that next. So we are only at the beginning here. Right now, you just see tissues on the portal. But we're very lucky that in another context and another future webinar, we're working with Evan Rosen and Melina Klausnitzer and other folks at the Broad. Evan is what I would say, the leader in all things fat. And so he thinks about that tissue in a way that nobody else does. And, he's been, passionate about building a resource for that. So we're very lucky to work with him to be funded to build a resource for mammalian adipose tissue. One of the core features of that is browsing cell abundance across adipose tissue. And so we've started to build that. And what's great about that is that you have like one disease, one sort of, proof of concept to how you would represent single cell data in the context of various conditions and what questions would you want to ask. And then we can take that as if we do that well, we do that for that community really well. We have a lot of work to there. They're generating a lot of other data types as well, but this is adipose tissue and it's incredibly relevant to CMD. But more importantly, some of the visualizations we're thinking about it, we have some early versions of it, and I thought I'd just share with you, some of the things we're thinking about there, which won't be new to you in terms of seeing them because you've seen them in papers. But what's nice is we're slowly laying the groundwork for how this may be integrated into our human genetics context. So I'll quickly show you guys a few snapshots of that. Here we go. So this is one data set, for example, that Evan and his team published a couple of years ago, 2022. And this is part of their single cell atlas of human subcutaneous adipose tissue. And, this is one data set, but we want to build integrated maps. We have a lot to do, but just gives you an idea of like how we are thinking about presenting this. And this is, not super novel, I would say, in terms of like, you haven't seen this in other resources. But what I wanted to do is just sort of bring you along for our journey. So for this, the first thing you want to lay out is like, what is the study? What is like, sort of orient the user to what was done, the different BMI states, the different donor characteristics in the data set. And this is just like one, view of the functionality. You can quickly see the lovely functionality here that, gives you a table, the summary of the different donor IDs. This is very specific to the data set. That's great. That just sort of orients you. But really what I want to talk about today, with you is like the visualization of the different cell types, excuse me. And so this is one start at, looking at the various clusters of adipose tissue across this particular data set and the UMAP plots associated, but then allowing you to query by this. If you want to query by sex, for example, and you see that here, you can see it change dynamically. This dynamically means it's already on. There's actually sex clusters. And you can see how that actually changes. You can also do it by BMI group, for example. There we go. It's a little slow because it's beta, but this is an example of how you can see the different states by cell type. And we're hoping that this is sort of the substrate by which we may integrate this in other resources, but also build resources like this for a specific tissue around adipose tissue. And for each of these, you can see the different cell types, clusters by totals, the specific tables, and all the data that goes into it, and the clusters below. What's also nice is what about gene expression, right? So same thing. So I'm just sort of walking you through this because this sort of models what you saw earlier in the Kyle's paper on islets, which is like present the distribution, the study design, the cell distribution, and marker genes. And so this is one place where we allow you to query, your favorite gene and see other ones and see how the distribution of gene expression changes across the different cell types. Again, we want to build this out more intuitively and also figure out how this corresponds back to the genetics, right? Because that's really the main mission. So the next sort of phase here is to bring a differential expression to this and also other, features that make this more user-friendly. But I just wanted to sort of tease you with the groundwork for eventually this will come into the knowledge portals over time in terms of functionality. Because ultimately, and this is, I think, my last slide, I think from this review, one of the things that really stuck out to me is like, this is the type of data that people are going to be generating. Disease, no disease, but also disease-associated variant is studied in that person, and you pulled else assay information out, you actually are conducting single cell experiments in these different states. So you can eventually actually visualize them, see them all together, and potentially actually make decisions about the gene, the cell type, the pathway, and the transcription factor if you pull this all together. And we hope that the resources that we're building and the infrastructure that we're doing will help in the data sets that we can then ingest and incorporate from the community, which are being generated at scale now, can be made more useful to others. So that's kind of where we're headed. And I wanted to just sort of describe that to you, because over the next couple months, you'll see a lot of changes on the portal. And frankly, we have to reach out to a new community of investigators, who do this work. That's why we're so thankful to be working with Evan and his team. And then we have Kyle, people who specialize in building these maps, a specific tissue, generating data in them. How do we make them accessible? That was the success of the original portal. Like, we were human geneticists, we worked with them, we knew how to do the analyses, but it takes a while to do this with judgment and taste. So if people have thoughts, or people are particularly passionate and excited about working this, we do invite you, because I think we're only as good as the people who want to use our resource. And so for me, if you want to contribute, I think that means you're interested in being a user. So that's what I wanted to share with you today. And I look forward to ASHG, because you'll see a lot of this stuff coming up on ASHG. And again, this whole endeavor is a massive community, and it's also a really cool, cross oceans, cross country team that meets really regularly. And I wanted to point out a new member of our team, Julie Jergens, who just joined us like less than a month ago, and she is going to be our manager of content and curation and representation. So we're really super happy to have her, because she'll help us tackle some of these new portal endeavors. So with that, I think I might end and take any questions or comments or feedback. I threw a lot of information out there today, so I apologize if it was a lot.
No, well, we have a question in the chat from Doug. Yeah. It was from a little while ago, so I'm not sure exactly what it refers to. Doug, maybe you could clarify. Oh, we might have to let Doug speak.
I'll let Doug speak.
I just promoted Doug to talk. So.
Okay, thanks.
Hi, Doug.
Hi. I was just marveling at this great integration they're doing as part of the AMP and systems biology. And there are a lot of portals, and my comment was there are a lot of portals that you support that don't enjoy the exclusive AMP funding, but come from other sources of funding, and just wondered if there will ever be a chance of integrating, like you've already done, across portals that aren't part of AMP.
That's a great suggestion, Doug, and a great point. And ironically, one of the major drivers of AMP SysBio is NIAMS, which also thinks about musculoskeletal disease, which is something that you and I have worked on together for several years now. So our hope, given what you just said, we have not just for CMD, we've been really blessed to have other communities like yourself want to have the same challenge and want to invest with us in building portals and generating new features and functionality, but also bringing in data. Our hope, and I think Jason and I agree on this, is that as part of this SysBio initiative, since we are part of it as investigators, we will be able to, with the blessing of the community, figure out how the infrastructure that SysBio builds is useful to other portals that we generated. So the answer is yes. A little more complicated, but I don't think as complicated as funding. I think funding is not the issue. It's more just like making sure the governance and everyone's comfortable with that. So that's our goal, Jason and me, and how we think about this. And so I hope that we can achieve that goal. Absolutely.
Great. Yeah, especially since one of the biologies of aging is inflammation. It's
absolutely
one of the fundamental ones. And you mentioned you have this aging portal that is not part of AMP, but is obviously intimately related to inflammation.
Totally. In fact, when we did this AMP SBI thing, that readout of all the resources, this is when I got to know the folks in AMP AD. And, they think about aging, obviously, and they have a longevity resource. And they were like, wait, you have an aging portal? And so as a result, we have a kind of a budding collaboration with Sage, Anna Greenwood, who I really enjoyed working with across this AMP SBI effort, to think about maybe trying to figure out those things would come together. Again, all in the beginning stages, but to your point, Doug, really a mission that, you know, I hold dearly in my heart to try and engage people in making these data integrated and accessible to many people. Evan, you have a question?
Evan, you should be able to chat now.
Yeah, can you hear me?
Yeah.
Okay, great. So yeah, the question is about the long-term plan. So you're building all this incredible infrastructure, and you know what's happened to some portals in the past. I'm thinking of the nuclear receptor signaling atlas that we used a lot for a while. It just fell into disrepair and was ultimately taken over by pirates. So what's the long-term plan here? Do you think industry is going to support this? I worry about the long-term you know, ability of the NIH to do this.
It's an incredibly good point, and it's something we worry about often. So I'll tell you, the way that we've tried to address it internally, because we can't control what people want to, we can't control our, you know, our funders because they have, that's their decision, and we're lucky to have funding we do now. But we control internally is trying to make software that is flexible to different scientific questions. That first really helps. Minimizing the burden of data storage is another thing, because the cost of these things, you know, you need to make them small and as much as possible. So that's why summary representations, pulling infrastructure, federation, pulling infrastructure from other places, not in reinventing the wheel. I'm saying things that sound really basic, but if you really adhere to those principles, then the portal doesn't fall into disrepair as much. So I think the reason that, you know, what we're doing with you, I didn't even know in the call until now, this is great. The reason we're doing MatKP with you, right? We're going to have that resource, but the fact is that some of that functionality can go into CMDKP is because there's similar scientific questions, but they are leveraging each other. They're different funding streams, and we're doing what's mandated for both, but we're trying to make it so that when one grant ends, we can actually support both, because adipose data is relevant to CMDKP, right? And some of the genetics that you want in MatKP are relevant to MatKP. Wait, I didn't say that right, but you know what I'm saying? So the beauty of data types that can span resources makes the resources fresh, and working with the community to make sure that the resources pivot to new scientific questions nimbly over time, I think does allow them to be more sustainable, but I don't know what the future holds. I do worry as a developer, you know, like we're making a new portal, right? The SBI, but it's not, hopefully if we do it right, it's not really a new portal. It will leverage, you know, not like rebuild infrastructure that the 10 amps have already built, but like find a way to lightly stitch them together and just create an infrastructure on top of them. So that's my hope. You know, that's what we are trying to work with the NIH. Our industry partners are amazing about giving us feedback. NIH is deeply, deeply invested in trying to help us with, you know, cross-cutting resources that talk to each other. So that's why we partner with the Common Fund. That's why we applied for that, because that brings those resources immediately together for us. So like we have gene expression, differential gene expression coming in from Common Fund, because that's a specialty of the Common Fund resources. That those results, we hope to make those accessible in our disease portals and vice versa. So there's a lot of, we look for ways to make things shared across, and that I think helps with sustainability, but it's a challenge. I hope that answered your question. Probably more than you wanted.
Thanks.
It's actually, these resources, these conversations are also part of the way that we do this, because people have ideas, and like Doug, you reminding me of that reminds me that I'll always keep that in mind when, and that gets you involved. So I think really for me, the reason I talk about these, you know, just give you guys sharing on many of this is that if you're interested, it creates opportunity in other areas we hadn't anticipated, which I think is the reason these resources can be successful, is you constantly are looking for the new opportunity to make them useful to somebody. So hopefully we can do that with you all giving us suggestions. Any more questions or comments?
It looks like we have everything answered in the Q&A.
Awesome. So I'm really hopeful. I think we're hoping to have a version of the MatKP for the spring of 2025, and that's going to be very exciting. You know, for folks of you who may not know this, in another setting, we are funded kindly from the NIDDK with another group led by Steve Parker, but Kyle's leading this to build a pancreas resource. Again, same concept, same challenges for the human pancreas. But again, the beauty of that is we're going to leverage stuff we've already done, serve that community and that scientific gap for the pancreas, community, but do that with infrastructure and flexible platforms and expertise from communities we've already worked with. And that makes us capable of doing this, I think. So there's going to be a lot of exciting things happening in the coming months. And I hope that we serve the community effectively. That's really our goal. So this is recorded and it'll be on our YouTube channel if you guys want to take a look at it. And if you have any questions or follow-up comments, so this made you think of other things, please, you know where to find us at all times. So thanks for your attention and comments. Any other comments before we close? I can give everybody back 10 minutes of their day because it's a beautiful one up here at least.
Thanks.
All right.
Thank you, everybody.
Take care.
Bye.